This one.
It's probably the most challenging one because it has the shades of color. The talk today is sort of a personal history in some respects, in that it's going to take you through my thoughts on how I try to understand the linkage between changes in the brain and aging. I come from a background in neuroscience. I've got some background in computational modeling and so on. But my main focus actually has been neuroscience and psychology. So I'm coming at this from sort of the traditional cognitive psych perspective. But as I've grown up, I started working with other colleagues in other fields, like in computer science in particular, but also neuroimaging and so on. And they've sort of crafted my thinking about how to conceptualize changes in the brain, how that impacts cognitive and behavioral functions as we age. So some of the stuff might be a bit too detailed. I'm hoping you'll get the gist of my talk as I go through it. I want to make sure this is interactive as well. So if there are questions as I go through this talk, please feel free to stop me and ask for clarification. The talk is divided up into three major sections. One is really sort of a theoretical overview of how I think about how the brain works. And that theoretical foundation you'll see revisited a couple times as the talk will proceed. And it provides a framework to understand some of the stuff I'm going to be talking about with respect to the empirical data, primarily that coming from the neuroimaging work that I've done. And there's a section then on mapping age-related changes in the brain having to do with the relationship between cognitive function and brain activity, mapped either with functional MRI or, in most of the cases here, actually PET measures of blood flow. And the idea that you can get these differences in maps that both relate to preserved performance, in quotes, with aging, as well as performance that's below that of younger subjects, or other words called impaired sometimes. Actually probably the last half of the talk really focuses in this last section on brain noise. And this has really helped, looking at brain noise has actually helped me to sort of reconceptualize a lot of things about how, not just the brain, but actually a lot of parts of the body are actually operating. And in the sense that we think about noise as a bad thing. We can think about noise in a radio, or noise on a computer, or noise in the room. We think about that as a bad and distracting thing. But in biology, and in particular in the brain, we're talking about a system that's highly nonlinear. And these nonlinear systems actually are enriched by having an optimal amount of noise. So it may very well be that a lot of what we see in aging, whether it be brain function or body function, is some change in this noise profile. So we'll get back to that in the latter half of the talk. In terms of how the brain is set up, one can conceptualize a continuum going from a situation on the right side. I'm right-left agnostic. So I do this and say, oh, this is left. You have an independent system where you have channels of processing that don't communicate with one another. So if you perturb a node in this system, I'll try and stick with it. Here, for example, you'll get an output. Preserve that, you perturb this node, you'll get a different output. On the other extreme, you've got a situation of a redundant system where everything contributes to any function. As a matter of fact, what ends up happening is that the repertoire of the system is quite reduced. So if you perturb a node in this redundant system, no matter which channel you hit, you'll get the same output. A redundant system is quite fault tolerant because you can eliminate any one of these nodes and you'll still be able to preserve that function. The problem is, since everything contributes to that function, it doesn't have a lot of variety in what it can do. So no matter what node you perturb, you're going to get the same thing all the time because it's completely redundant. In the independent system, you perturb a node, you get rid of a node, you've lost that function. So it has almost no fault tolerance. It so happens that the brain is actually in between that range. It's called a degenerate system mathematically. It's not a personality quirk, it's just that's what it's called. And what that suggests is that each node contributes to overlapping functions. So you have a high degree of local connections here, and more sparse, more distally. So there's a gradation in functions you go across the system. That provides is an optimal amount of variety in terms of what it can do, but also fault tolerance. And you can further define that as this system actually has maximum complexity. Complexity is actually, in terms of mathematical definition, as being the optimal ability to both differentiate and integrate information. And that is where the brain sits, in that sort of sweet spot called the degenerate system, degenerate architecture, having maximum complexity. So what does that mean in terms of understanding how the system is then functionally organized? Can we then look at the system as an independent system and say, if I see this node active, that means that that's the only function that's being manifested. What it actually suggests, if it's in fact true that the brain is degenerate, that different parts of the brain can contribute to overlapping functions. And that's this idea of what's called a neural context. That the functional relevance of an activity change in one part of the brain depends on the activity of other brain areas. So there's a broader context within which brain networks operate. The bottom part is a quote from a paper published a while back, talking about neuroimaging data in particular. And just saying that the important factor when you're looking at imaging studies, for instance, is not that a particular event occurred at a particular site, but rather under what neural context did that occur. In other words, what was the rest of the brain doing when that part of the brain was active? The cartoon on the far side of the slide, that would be left for me, shows you the idea of neural context. You'll notice that there's 4 nodes here that are engaged in both of these functional networks. But on the top network, you've got these more anterior nodes interacting. On the bottom set, you've got these more post-ventral nodes interacting. Same 4 set of nodes, but by virtue of the fact that they're interacting with different systems, i.e. the context is different, the emergent function therefore differs as well. So to appreciate the function, you have to have to look at the entire context within which a particular part of the brain is engaged. It makes it somewhat complicated, though, when you think about how things change as we age. Because it might be the case that this context actually changes as well. So this could be the normal situation, or we'll call it the young adult. In aging, we may have a situation where some part of that network shows some degeneration. For example, let's say white matter degenerates somewhat, so that the integrity of these pink-colored nodes is reduced. So what ends up happening is you've got a new set of nodes that are engaged, trying to maintain this original function. So you have this scenario here. And the question is, what is the functional consequence of that? That change in how these areas are interacting, the change in context, if you will. Okay. So it's really going from thinking about, for example, one of the most popular areas to talk about in aging is the hippocampus, for instance. Hippocampus shows some structural change, and therefore when you see cognitive changes, it must be because the hippocampus is messed up. It's actually very hard to show that in normal aging. But what you do find time and time again is that there's a network reconfiguration. And the point then being that there actually is a context change in normal aging. And that's really what's happening. The whole system is shifting somehow as we age. So these physical and anatomical and physiological characteristics, this idea of degeneracy, is present throughout life. Those principles are there in the moment you're born, in the moment you pass away. The brain is also quite malleable, as you probably know. It was originally thought that plasticity, which is something that allows the brain to change how it's wired, was something that was limited to infants and young children and young adults, but actually it's there throughout life. Varying degrees, of course, but it was there throughout life, which means that networks can reconfigure themselves as we learn. but there'll be functional consequences for that. So the slide I showed you prior, where you have a change in those anterior nodes, is going to be the attempt of the brain to change how it's going to function, to recover, if you will. There will be an implication behaviorally. And so you'll see that as this idea of impaired versus preserved performance. I put that in quotes because it really depends on what your baseline is, whether it's impaired or not. And usually our metric is whether or not there's a change in behavior relative to young subjects. So let's go through some examples. In the first situation, perceptual matching task, we have a situation where you get different patterns of activity in the brain between young and old individuals, but the performance in this task is actually the same. In a latter study, you've got a memory task for faces, and you have different patterns of activity between young and old individuals as well. And the question is, how do you link those two things up? This is a very old study done by Cheryl Grady when she was still at the NIH in Washington, or actually Bethesda, looking at the activation from people looking at faces or looking at spatial locations. And what the maps show you are the areas in red are areas where there was more activity in this task in older subjects relative to young, whereas those in yellow are where there was more activity in, sorry, other way around. Red was higher in young than old, yellow was higher in old than young. And again, despite the fact that performance in this task was pretty much the same, some slight change in reaction time, but not significant, and accuracy was identical in this task. So in the face of identical performance, you actually get different patterns of activation in this task. You'll notice the areas in yellow, more anterior, are actually sending into the frontal lobe. We can take these data and look at them in a bit more detail and actually make a path analysis, structure equation model from these data to see how these areas are actually influencing one another. And what you find when you do that, the top are some young subjects doing the face matching task. And you find when you put this into a path analysis and see what are the effective connections of what's called looking at directional effects. You see that the younger subjects, a very nice flow, if you will, of information from the posterior areas into frontal cortex, but not much coming back, if anything. Older subjects, you have this very strong loop set up now between the ventral areas going into frontal cortex and then heading back to area 19. That may, in fact, account for the fact that you have more activation in frontal cortex in the elderly subjects. And just as a comparison, if you do the same analysis and task in individuals with early Alzheimer's disease, you see a complete breakdown in this anterior-posterior set of interactions. Okay. So again, same performance, different set of areas interacting. This is a map from the face encoding and recognition study. It's a rather complicated color scheme. You can see there's actually less activation in the older subjects in a lot of these comparisons between the baseline task and the encoding or recognition task. So for example, if you look at the frontal lobes during encoding tasks in young subjects, you got a massive activation of frontal lobes, much less so in the elderly. And the same thing happens in recognition. You got less activation of frontal lobes in the elderly during recognition. In this case, the performance is worse in the old subjects. And also, this is just showing you now pairwise correlations between two different parts of the brain. Just showing that not only the activation is different, but again, the areas that are interacting are also different. How they're interacting is different. So for example, if you do a correlation between the hippocampus and the anterior cingulate cortex, very strongly connected in young subjects, basically zero in the older subjects. Conversely, if you look at the hippocampus with its contralateral partner, right and left hippocampus, you see a respectable correlation in the young subject was incredibly strong in the old subjects, almost suggesting that there's a completely different system that's engaged. And it's interacting like crazy, but it's not doing a very good job of it because you don't remember what the face was. Okay. Interesting. What does it mean? We'll get there. Now, when I started getting into this game, I was kind of concerned about the fact that a lot of these tasks had a number of levels of cognitive complexity to them. And the hope was that I could come up with a task that really sort of looked at some sort of basal function. So the task we picked was this very simple perceptual matching task. This is a collaboration actually with Alison Sekula and Pat Bennett, who at that time were at UT and now are at McMaster. The task is, you basically have two sine wave ratings. And your task is to indicate which of the two sine wave ratings have a higher frequency. Which one has more bars, essentially. And you have a variable delay of either 500 milliseconds or 4,000 milliseconds between the two intervals. So it's a very simple perceptual memory task. It's hard to think about how you would encode this semantically, because you can't count the bars because there's not enough bars to do that. And you can't really attribute, like say, that looks like a bird, for instance. It's hard to do. You could, but it's not going to work very well. You look at performance in terms of the difference between the gradings that's necessary to achieve 80% accuracy. This is plotted as threshold, but the thresholds are not different between young and old subjects. So in terms of group differences, they're actually matched. But what you do find, there's a delay effect, so the threshold gets higher when you increase the delay, but there's no age effect. If you look at the areas that are engaged in doing this task, you find quite different sets of networks that are engaged. On the top, you see a set of regions that are interacting and supporting good performance in this task in young subjects. And these areas include ventral frontal, inferior temporal, and occipital cortices. If you look at those same areas in these healthy elderly individuals, you find that those functional connections, these are correlations, go down. And instead, what seems to support performance in this task in the elderly subjects is the interaction between a dorsal part of frontal cortex and the hippocampus. Remember, again, this is a very simple perceptual memory task, not typically one you would associate with hippocampal function, and yet, in the old subjects, they use this circuit to support good memory performance. So the question then is, what's the consequence if now we scale the task up and add more cognitive complexity? Can they in fact switch the networks and continue to use the hippocampal system to encode more complex stimuli, or does it get messed up and that's what's underlying memory deficits that you see later on? Let's check that out. So a study that Cheryl did as a follow-up to that looked at higher order memory tasks. And this involved either words or pictures. The Snodgrass van der Waals line drawings. We found, as other people have found, that when you do this task in young subjects, that the successful memory encoded actually related to how much hippocampal activation was present during the encoding part of the task. The question is, does this also happen in elderly? This is performance in the task. Hits minus false alarms. There's no statistical difference. There's a somewhat reduced accuracy for words. For objects, there's no difference between the two groups in terms of accuracy of memory performance. I think there are about 15 subjects per group, so it could be a power issue here as well. But for the purposes of today, we'll say there's really no statistical difference in performance between the two groups. If you look at what brain areas considered across the entire brain now relate to hippocampal activity and to performance, you get these kind of funky maps. So what I'm showing you here are the areas that are both related to hippocampal function, so functioning connected to the hippocampus, but also are correlated with behavior, accuracy. But they're not correlated the same way in both groups. First of all, the spatial pattern is very distinct for the young subjects. The hippocampal seat is down to here, where the arrow is. The areas that are positively connected or positively interacting or functionally connected to the hippocampus are the ones that are in yellow. So you got middle frontal cortices as well as the other parts of the temporal cortex. On the far part of the slide, you see the correlation with the seat as well as with behavior is quite high in the young subjects. But this pattern shows virtually no correlation with the hippocampus, nor does it show any correlation with behavior in the older subjects. But this pattern does in the older subjects. This pattern has strong functional connections with the hippocampus in the more dorsal parts of frontal cortex, which are the same areas that we showed you earlier in the perceptual memory task, and are correlated with the hippocampus and nicely positively correlated with behavior in this particular case. So again, this is where the contexting comes in now, because we have the same part of the brain, putatively, that being the hippocampus. By virtue of the fact that it's correlated with different parts of the system, it actually differentially supports behavior. In one group, there's no relation to behavior at all. In the other group, is actually strongly related to behavior. So the change in that context actually allows these systems to reorganize and support behavior differentially as the person ages. At least that's the interpretation we're giving. Interesting. That's what I just said, actually. Okay, so it seems like we have a fairly compelling story emerging. We're showing age-related changes in brain function that relate to cognitive performance. And we know that there are structural changes in the brain as we age. There's some volume reductions, there's some weight matter changes as well. And it seems to be the case that there's something that happens in the brain to, at least in most of us, maintain a relatively stable cognitive performance as we grow older. Question is that a system level change, or is that the whole brain? So for example, I showed you some instances where you have frontal and hippocampal areas changing interactions. Is that all there is to normal aging? That in order to preserve cognitive function across a broader range of tasks, we, those of us that are getting older, will tend to engage dorsal frontal cortex and hippocampus as we get older. The answer to that question is no. It's actually not true. Because you'll see changes happening across a lot of dimensions with different parts of the brain. So areas that support motor function will also change. Just actually reaching behaviors will change. Areas supporting that. So it's like the whole brain's changing now. So what's going on? Structural changes, there's functional changes. They don't seem to be quite as specific as we'd hope they would be. There are commonalities, of course, otherwise, as you get older, the whole world would change dramatically. It doesn't really change that much, except in the winter, I think. But it's more than an individual functional network. It seems to be something more globally happening. What might that be? So the question is there something more general? Is there a general principle that may help us understand these sort of global changes that are happening to our brains across the lifespan, but in particular as we age? And that's where we get into this notion about brain noise. So, cheers. Several years ago, I was at a talk on auditory function in aging. And the presenter was actually out of Scarborough. Presented quite a compelling series of experiments that ranged from very simple auditory perceptual tasks to much more complicated auditory cognitive tasks, like auditory objects, auditory memory, and so on. And across this array of experiments, there was no consistent theory that could link all the changes that one saw. So in some tasks you would see age-related changes, in tasks you would not see age-related changes. So it wasn't as if, for example, you have just peripheral stuff messed up in the cochlea and that would account for everything. And it wasn't only central as well. It was kind of like, again, there was a lot of stuff happening that didn't seem to have a unifying principle. So I heard some stuff around the time on a phenomenon called stochastic resonance, which I'll explain in a second, which has to do with noise and the putatively beneficial effects of noise in nonlinear systems. So I thought, well, maybe that's what's going on. Maybe let's take a look at that phenomenon in aging. So I started perusing the literature and see, well, what do we know about variability in noise in aging? This is a review paper that was published in Trends in Neuroscience several years back, looking at just variability in behavior with age. This is intra-individual standard deviation in a simple reaction time task. Cross-sectional study across a gazillion individuals. And you find, not surprisingly, that variability in reaction times is quite high in kids, stabilizes somewhat in early adulthood, and then it kind of fans out. So instead of being an inverted U, it actually kind of goes. When you start digging even deeper, looking at what are the different sources of noise in the brain. This is quite a good paper from Faisal, Selin, and Wolpert. Focused primarily on motor function, but this is a general review is quite good if you want to take a look at it. And this is just showing you that the brain is an imperfect system. It's not a computer. It's a biological system. So it has noise. It has noise throughout the entire axis. If you look at someone trying to catch a ball, there's noise in the sensory apparatus in the retina. There's noise in the transmission of the retinal impulse into the central part of the nervous system, up and through here. There'll be noise internally between the different networks that are interacting because of the slop in the channels. So you have imprecision in terms of firing, imprecision in terms of frontal release of transmitters, for instance. There's also slop in the effector system, going from the spinal cord to the motor, to the muscle, as well as synaptic noise, again having to do with imprecision and channel release. The question is that just a consequence of biology being imprecise, or is there actually a functionally relevant reason for that? And that article, as well as the I'm going to tell you now, suggests that this noise actually is adaptive. That we need that to a certain degree for our brains to function optimally. This is stochastic resonance. I'm going to show you right now. So we'll do a pretend experiment where we have a very weak signal. We're trying to detect the signal. And we're going to plot that detection as a probability of something being detected for a given level of noise. If your system is linear, what you'll find is that as you increase noise, the likelihood you're going to detect that signal is going to go down. With a nonlinear system, you see this rather nonlinear function, that as you increase noise, the system will go from being similar to a linear system to actually doing better than a linear system, and then start falling off as you increase noise too much. We can do this experiment even better with an actual example. This is a image on this side of the screen. You see that there's a lot of noise and it's very hard to detect that image at all. If you start adding in random noise, random white noise to the image, you see that as you scale up the noise, the image emerges here optimally. You can kind of see it here. Here it's not easy to see. And at that point it starts disappearing. So again, it's just linear adding noise, but it's a nonlinear function in terms of your detection, which means you are a nonlinear system. Congratulations. What I also found was that people know this intuitively. At least some people know this, engineers for sure. So you can develop things that help people with peripheral neuropathy, for instance, have a hard time with maintaining balance and equilibrium. This is an example where you have somebody standing on a sway platform. And standing on these little vibrating insoles. And without any noise in these insoles, you just turn them on and they're imperceptibly vibrating. Without noise, you see that their sway pattern is quite wide. But when you turn the noise on, you see that it actually reduces. So they're better able to compensate for the change in the platform. And this group actually went out to market it and create vibrating socks, which are probably not very good in the winter. Step in a puddle and say, oh.
Batteries included?
This is big battery pack. But yeah, so it has actually an adaptive function. You can see how that could be used. And as a matter of fact, in hearing aids now, in cochlear implants, they're also now adding noise to these systems to help with detection, particularly in cochlear implants, but also now in hearing aids. So there's something to this noise thing that makes it seem like we should look at it a bit more. You remember at the beginning of the talk, I mentioned this idea that the brain is a complex system, that it has this capacity to both differentiate and integrate information. It's a nonlinear system. And nonlinear systems, complex systems, generate more noise. If you measure systems like the one at the bottom, that's basically a lattice, because it's completely interconnected, it's redundant. If you measured activity from that system, it's very deterministic. Idealized, for example, here is just a sine wave, because it only does one thing, right? This repertoire is very reduced. As you make the system more degenerate through this architecture, you find that the signal it generates becomes more complex, has more noise. It has more information in it. The system has a higher capacity to represent information because it can both differentiate and integrate optimally, which is how you get a system that's maximum information representation. So a system with a complex structure will generate noisier signals. We can map that noise onto a notion of repertoire, or states, or configurations, or knowledge, if you will. Here's a really simple example of a very, very simple neural network model with 3 nodes. They can't do too much. But without noise, you perturb, these are nodes that just oscillate. So when you turn them on, they just do this. Without noise, you turn the system on, they oscillate a bit, and then they kind of flatline. And you can plot that behavior in what's called the phase diagram, which is what you see in the bottom. And that tells you the space that this 3-dimensional system occupies. So it's a nice space. And you see the trajectory that this system is doing as it goes to what's called a fixed point. And the fixed points where the system's flatline, so it's also called an attractor in the language of dynamical systems. On the other side of the screen, we have the same 3 nodes, but we've added some noise for each one of the nodes. Basically made them 3 different neurons now. What we see now is that the space that the system occupies is pretty much the same. It's the same 3-dimensional space. But the exploration of that space is much more thorough. When you perturb that system now, you do the same kind of plot, you see that it takes a sweet time now getting to that attractor. It's going around and exploring completely that phase space, exploring its repertoire. What are the possible configurations that can be occupied by the system in the presence of noise? We can explore that further in this platform we're developing now called the virtual brain. I was mentioned very briefly in my introduction. What it allows you to do is take data from human neuroimaging experiments and use that as the basis to construct large-scale models. So I can take anybody's brain, essentially, put you in a scanner, get information on your brain's connection profile and functional profile, and then generate a virtual model of your brain activity. That's the idea behind the virtual brain. So I put it in the computer, and I can generate activity I would measure if I was to stick electrodes in your brain, which I don't want to do because it hurts, at least the first part. Or I can generate things that I would measure. I put a cap on your head, measure EEG, or if I put you back in the scanner, I can generate fMRI signals with the model as well. The nice thing is I can actually then take these models and see what are the critical parameters in the model that allowed the model to represent the data that I collected. So going back and forth between the model and the data. This is a complicated slide. I'm just going to walk you through it. This is telling you something very critical about the behavior of the system now, and where it sits in terms of noise and connections. So in the first column here, what I'm showing you is I have a full brain model where I've taken, I think in this case it was 10 different individuals, and did an average connection matrix. And then just at each one of the nodes, just put in these simple oscillators that I showed you earlier. And I did things like scale how strongly they're connected to one another, and also how much noise there was in the system. And when I do that, I can map that model back to the empirical data again and see how well do they correlate with one another. So as I scale the connection strength, and I correlate that with the empirical data, that's what this top plot is showing you. That with a low coupling strength, low connection strength, there's not much correlation. As I ramp up the connections, you see that at some point the connections become stronger, or correlation of empirical data, I should say, becomes stronger. But if I make the connections too strong, then the correlation drops off again. Tell me there's actually a sweet spot in terms of the connection strength, in terms of being able to go back to the empirical data. When you do analysis of what's happening in this region where the correlations are peaking, you can look at this space again to see what does it look like in terms of the number of attractors that are in that space. What are the number of states that this model can actually take? What you find happening is it transitions from that very simple single attractor to multiple attractors. So again, going back to the example before, you go from having a single point that everything kind of converges to having multiple different configurations that are possible. Okay. But if you go too far, then it collapses back into a single space again. So some optimal space in which the mapping of connection strength and noise allows there to be this complete exploration of potential functional configurations, the repertoire of the system, dynamic repertoire of the system. If I then do something like introduce visual stimuli to this model, say, okay, here's a picture, and see how fast does the model react to that picture. That's what's plotted in this column over here. Okay. If I do it here, in this region, in the sweet spot, you see that the reaction is actually stronger. If I do it here, the reaction actually goes down because I'm just at the edge where it's going to collapse. Okay. Sorry, faster here. I don't have a C, but if you go over here, there actually would be no response to that stimulus as well. So again, we're getting optimal behavior responses in the system as we get closer to this sweet spot here, this point of maximum correlation. So the way to think about this is this kind of like, who plays tennis? 1%. Squash, racquetball, volleyball, anything like that, no? Okay, let's think of a different one. Who watches tennis? When you see people playing tennis, when they're waiting for a serve, they're not standing still. Right? They're kind of just going back and forth, waiting for the serve to come. If they move too much, then the serve comes and they're in the wrong location. They've screwed up. If they're not moving at all, they probably tear a muscle, or they won't get to the serve. But if you're actually moving a little bit, exploring possibilities, you will actually optimally react to that serve. And that's the same kind of analogy I think what's happening with the brain. If there's enough internal noise that allows the system to respond optimally to a perturbation. So the model tells us that there's something about anatomy that's important. I didn't show you this, but if you scramble the anatomy, keep the number of connections the same, but change who's connected to what, this whole thing collapses. The strength is important. It's kind of obvious that if you're not connected, you can't hear what you're saying. And the last thing, that noise is also important. That these three things seem to set up a collection of potential states that reflect the brain's capacity to respond, the repertoire of the system. So if brain noise reflects the brain's repertoire, it should change then with maturation. It seems to be a reasonable hypothesis. So one of the questions could be is that can developmental changes be partly accounted for by changes in the noise structure? We know that kids change what they do as they mature. Sometimes for better, sometimes for worse. The question is part of that at least reflecting what is changing noise structure? So the next, I'm going to be going through some empirical data. There's two hypotheses we're testing in these empirical data. One is that early maturation may actually lead to an increase of internal noise in the brain, whereas the behavior of the aging nervous system is consistent with a reduction of noise, internal noise, brain noise. So the first experiment we did on this looked at kids from 8 to 15 and their response to faces in a simple memory task, a one-back task. So does this face match the one you saw previously? These are the evoked potentials, average evoked potentials within each of the age groups. This is not surprising in terms of the patterns you see here. I'll just go through just so you know what the typical response is. So in the youngest kids, when you look at a face, you get a very strong, huge response. It's actually even larger in infants. And in case you get asked me later, it has nothing to do with skull thickness. It just basically is the way the system is. That's about it, though. You have a single massive response, and that's all. As the kids grow, as with age, I should say, you see that the amplitude of that response decreases, and the latency becomes shorter. So it becomes faster, and it's hard to see because I matched the amplitudes, but there's a more multi-componential response. So by the time you get to the adults, you have the typical P1, N1, P2 complex that you will hear about in visually evoked potential literature. So youngest kids, huge amplitude, slower, older kids, lower amplitude, and faster. Okay. Is there a change in variability in these data? Within subject variability, how they respond trial to trial in these data. So how do we measure that? Well, from basic statistics, we know that if you have more variance, that means that there's probably more noise in the system as well. So a very simple way to measure noise in the system is looking at something like a principal components analysis, which is very sensitive to variance. So the more variance you have, the more components you have. The higher the space is, the more principal components you need to capture the variability of that system. So you can do something like, for example, look at how many components do you need to capture 90% of the variance. And that number of components is the number of dimensions you need. So therefore, again, more noise, more dimensions to capture the variance. The second thing you can look at is not only trial variability, but also variability across time. So in a response, how variable is the response? You remember, for example, in the kids, the very youngest kids, we had a single massive response. And that's actually pretty much what happens across all trials, which means that across time, there's not much information there. But you can imagine as the signal becomes more jagged, there's more information in that time series. So we can look at principal components analysis and use a measure called multiscale entropy. Entropy here, you can think of entropy in the same way you think about information. So more entropy, more information. This is the algorithm that's used for MSc. I'm not going to get into great detail, but just to kind of give you a feel for it. What you do is you measure the entropy of the original time series, how much information there is, and then downsample that and do it again. So look at information as a function of time scale, or if you will, frequency. That's not exactly frequency. For systems that have high information content, you'll see that the entropy stays pretty much stable across different time samples. For systems that are completely random, however, or very deterministic, you'll see that entropy drops off as you start downsampling the time series. Because in a complex system, information is represented across a number of different scales. You can capture that with this measure. Okay. And this is just a toy example, just showing you what I just told you. Is that here you have a complex signal, here you have a very deterministic signal, and the entropy for this complex signal is high across all scales, whereas for the deterministic signal, it's down there no matter what you do. Okay. So now let's look at brain noise in these kids. This is the results from the principal components analysis. How many dimensions do you need to capture the equivalent amount of variance in the group? Kids in the top, adults in the bottom, other age groups in between. The hotter the colors, the higher the dimensionality you need, the more noise there is in the group. And you see, from top to bottom, that noise increases with maturation. You also see, going from left to right, your left to right, that there's a drop in this dimensionality. And the drop has to do with whether you're pre or post stimulus. And that drop in dimensionality is actually consistent with a very deterministic system, one that responds, boom, to a stimulus, and always the same way. Okay. If we look at the entropy measure, multiscale entropy, now what I'm plotting here is the entropy measure for a single electrode. You have to trust me that this is pretty much the same across other channels that we measured with the EEG cap. That across all scales, from the original scale to the most coarse grained scale, entropy is highest in the adults, and the kids fall in ordinally below that. The maximum difference is actually at this middle scale here, but pretty much across all scales, entropy is highest in the adults, lowest in the kids. Brain noise is highest in the adults, lowest in the kids. So that's kind of cool. We can ask another question here and say, well, we have a task that they're actually performing. They're pressing a button. They're remembering whether the space was one they saw before or not. Let's correlate this with behavior and see if there's any relationship between performance and these measures. I wouldn't have said that if there was not a correlation. What you find, if you look at mean reaction time, there's no correlation with these measures of brain noise or variability. If you look at the stability of reaction time, coefficient of variation of reaction time, you find a negative correlation between the stability of the response and brain noise. In other words, the more brain noise you have, the more stable the response is. So it's shown on the top. Okay. You can look at the area under the curve for the MSc. plot, or the dimensionality estimation from principal components analysis. And again, you find a negative correlation. This is just the correlation at occipital channel. The correlation negative 0.05, and that's the upper and lower bounds of the confidence interval. Accuracy is a positive correlation. So the more noise you have, the more accurate you are in doing this task. Shown down here. There's definitely a correlation with behavior. As a matter of fact, if you regress out age, control for age, these effects stay there. They get somewhat weaker, but they maintain themselves, suggesting that there's something fundamental about this brain noise and its capacity to support good performance in this task. Let's now go to the other end of the spectrum and look at older adults. I'm going to go through two data sets, same analysis, just to show you that the results are consistent. The first one has three groups: young, middle-aged, and elderly. It's an EEG task. It's looking at bandpass filtered white noise, which is basically the same task I showed you a long time ago on perceptual memory, with some slight variations. The second task has only two groups, but it's an MEG data set. For those of you who don't know, MEG is kind of similar to EEG, but instead of measuring electrical activity, it measures magnetic fluctuations. So it's a very similar technology. And the task was looked at both auditory and visual responses. And what we did was looked at the entropy measures again, MSE, in this task. This is from the EEG data, and we have young subjects in the red, middle-aged in blue, and old subjects in green. And what you find is that unlike in the kids where you just have this sort of DC change, there actually is a dependency on time scale in terms of the age-related effects. So if in the fine scale, which is the original data set, very fine temporal scales, you actually have higher information, higher noise, higher entropy in the older subjects than the young subjects. As you go out to more coarse scales, the lines cross, and you get lower entropy information in the older subjects and correspondingly higher in the younger subjects. So the age effect is dependent on time scale. Is there a question? Stretching. MEG data set, the scale is actually the same. I just expanded the graph somewhat. Young subjects are solid lines, older subjects are dashed lines, same effect. That at the fine scales, you have more variability in the old subjects. As you go to coarser scales, slower time scales, the lines cross. So there's a temporal dependence, again, in terms of the age-related changes. Somewhat more complicated than the kids, but nevertheless, interesting differences. Now, the first part of my talk focused on the idea that brains operate as a network. What I've showed you thus far with the noise exploration is responses at a single region. But we know that the responses that you get from a particular part of the brain are dependent on what's happening in that part of the brain, but also what's happening with other parts of the brain. So there's both a local and distributed component to the signal you measure in a particular part of the brain. If the information that we gather by looking at entropy is reflecting that, then we should be able to parse the signal into that which is reflecting local dynamics and that which is reflecting the network level dynamics or network level noise. So we can actually then parse the total signal, this is total here, to that which is unique to the local dynamics, and that which is dependent upon the interactions between different parts of the brain. Local and distributed entropy or local distributed noise. That's whichever the case. These next slides are a bit busy, but you'll see why I'm showing you this. These are all the different sources that were measured with the EEG channels. There are 131 sources here. But the take-home message is fairly easy to digest from this. It is lunch after all. What you have in the red are the connections that show decreased entropy with age, whereas the ones in the blue are the ones that show increased entropy with age, increased interactions that had increased noise, distributed noise. And in the EEG data, what you see happening is actually the decrease involves the connections between hemispheres, whereas the increases are actually within hemisphere, primarily. This is not thresholded at all, so I want to show you the entire data set. There's no, you can run statistics on it, which I have, but these are basically everything that changed. So there's a change in distributed entropy, but it's primarily like the long distance connections are the ones that are going down the most. What also happens in this case is that local entropy, that which has to do with local dynamics, goes up with age. If you look at the MEG data set, it's even more striking. That the predominant change, again, in terms of decreased distributed entropy, has to do with the cross-hemispheric interactions, the long-distance connections. The information conveyed through those connections goes down with age, and you have an increase of local entropy, suggesting that these age-related changes are both temporally and spatially dependent. Last example, just to kind of hammer that point home, looks at fMRI data. You can get the details from the paper if you like. Basically, these are all healthy individuals. They're also happy. And what we looked at, instead of looking at simply the activation levels in the fMRI time series, is you looked at the variation of that. We can't really do the MSE measure on these data because the time series is too short, but you can look at the standard deviation of the bold response. Okay? When you look at the differences in the means between as a function of age, you do see that these areas in blue go down with age. So there's a negative correlation between the activity in these areas and age here. Even though it's mostly positive there, it's a negative weight swing on that side. If you look at the standard deviation, first of all, you find that the clustering is much tighter. So the age effect is actually stronger if you look at variability as opposed to looking at just mean changes. What you also find is that there are both increases and decreases of variability as people age in both. So the areas in blue are the ones that are going down in terms of the variance, whereas the areas that are in red are the ones that are going up with variance. This is basically the same general message that we had with the MEG and EEG data set, that the effect of age on information processing depends on space and time. We can also correlate these changes with behavior, and you find that, generally speaking, those areas that go down with age are also the ones that are maximally correlated with behavior. So if they go down too much, you actually become less stable in terms of your reaction time in these posterior medial parts of the brain. So they're negatively correlated with the coefficient variation in reaction time. Those are also the ones that are going down with age. So as they go down too much, you become more variable in terms of your response. So what is this noise stuff actually telling us then? First of all, noise changes across the lifespan. It's somewhat obvious that part of that, especially early in life, reflects the changing architecture. So you get differences in myelination, you get pruning that happens with maturation. Some of it has to do with experience. As you learn stuff, you're going to change configurations, you're going to increase repertoire. Learning languages, PhD, defenses, things like that, dissertations, you'll get different wiring. The changes early in life are quite global. We have gone back and looked at infants, and this trend I showed you with the kids from 8 to 15 is even more pronounced in kids from 1 month to 5 years. In adulthood, the changes are very scale specific. And this change is reflecting the change in the space-time structure of the brain. It's kind of a difficult concept to grasp, but just keep in mind that we're talking about a very complex system of interacting components. So it's not just what's active or when it's active, but what and when and where these things actually engage. The analogy I like to use, which I apologize for those who have heard this before, it's kind of thinking about a symphony, an orchestra. When you hear the tuba, or the drums, or the violins, in isolation, it makes absolutely no sense. When you put it all together, that flow, that variation in space and time, is what results in the symphony. And the same thing happens in the brain. It's not a new idea. You can go back and get reviews. This is actually published in 1966. A smart aleck grad student pointed this out to me. So this is not a new idea. See? Okay. Take-home message. So I started talking about networks and the idea of neural context, and that to really understand function of a particular region, you have to really put that function, that region back into the broader neural context within which that area exists. And that context can change as a function of experience. It can also change, obviously, in the case of disease processes as well, which is another topic. Most of my talk was noisy, so hence most of the devotion to noise here. It seems to be the case that actually a certain amount of noise in the brain is quite important for optimal function. And that really gets us to this idea of the fact that you have a nonlinear system in your head. These nonlinearities you can actually measure in other parts of the body quite well. As a matter of fact, the development of that technique they measure of MSE was applied originally to heart rate. And that, in fact, does show the same kind of profile. As a matter of fact, if you don't have optimal variability in heart rate, you die. So it's quite important to have variability in your heart rate as well. It changes with maturation. It is scale dependent in aging. That's the really important thing, is that time and space are quite important things to consider. It makes it more difficult to really conceptualize this. But again, if you go back to the symphony analogy, it makes intuitive sense that what and where things are happening in the brain, when and where, is quite important to keep in mind. And it's also probably quite useful in thinking about what happens in different clinical conditions. And in fact, we can talk about that if you like. And this idea of noise as enabling exploration suggests that what that means is that the brain is in a constant state of exploring what is possible. What can I do at this point in time? With that, I'll thank you for your attention. Take any questions? Come back.
Thank you very much. In your cohort of older adults, and I gather you were studying healthy individuals across the lifespan. In any cases in your studies, was there enough time to see if any of those adults were in fact undergoing negative change, whether from like MCI, mild cognitive impairment, or were you able, did you see, I guess you were not looking to capture
Yeah.
But that's like another phase of what could be studied.
Definitely. We didn't do exactly that, but I can give you some sort of ancillary anecdotes that will help, I guess, get to some of what you're asking about. The data I showed you with these different curves, those are group averages. What you can do is go back and look at each individual's curve. And what you find is that if you map those curves back to how people are doing on the task, if you take old subjects, if their curve looks more like young subjects, they actually do worse in the task. And conversely, you can have young subjects who look like old subjects, and they actually do worse in the task as well. So it's almost as if you do need this transition in terms of how your brain's set up to maintain optimal performance. There's another study we did quite a while back looking at, again, differences in brain activation patterns in a memory task for old subjects compared to young adults. And what we found was that the old subjects that showed patterns of activation similar to young subjects were the ones that actually did worse in the task. Something about changing your brain pattern that actually seems to be an adaptive way of aging and help. So we're getting at that, and that the question really you're asking really requires us to do a longitudinal study, which we haven't done.
I'm interested in the clinical significance of this work, and I'm thinking, of course, of the tennis player, the sweet spot. And are you talking about perhaps trying to get people to that sweet spot? I mean, is that what you would see happening, that you realize you're not within that, and how do you actually encourage that?
Yeah, so when I've talked to my clinical colleagues about these data, or actually working with them on some of these, they started measuring, applying the same measures to their clinical cohort. In schizophrenia, what we tend to find happening is actually there's probably too much noise, at least for some parts of the brain. What that means is if you have too much noise, you never actually get to that part. You get pushed into a different location. Your response is incorrect because you're moving to much longer baseline, using tennis analogy. In stroke, in TBI, and likely MCI, it looks like, there actually ends up being reduced variability in some brain areas. In depression, it seems to be regionally dependent. So again, there's a spatial scale dependency. It looks like things like stimulation, like deep brain stimulation, for example, what that actually seems to do is actually increase the noise to an optimal level. So getting into that sweet spot. If we can actually do that through therapeutic, through non-invasive therapeutic measures, that would be good. And use that as a measure to see if we can get there. The nice thing about MSE, for instance, is you don't need to acquire a lot of data to actually get the measure. You can do it in a single trial data. You need a long time series, but you can do it in real time and get that measure. So we're looking at that in some schizophrenic subjects actually right now to see if we can modulate noise in real time.
I have no science background, so this may be a simplistic question, but is there any evidence that either dementia or Alzheimer's lead or result from lack of adaptation in this way?
Well, that's a really good question. I don't know the definitive answer to that, but again, we'll just talk about some anecdotes. So I think there are things that are neuroprotective for Alzheimer's disease, which tend to be related to things that will increase your brain's capacity. So the one that people always cite is things like education, for instance. The more educational background you have, the more that you are protected from the degeneration from Alzheimer's disease. You still get Alzheimer's disease. You can essentially last longer for that. Exercise, for example, is another one. And we've done some studies looking at exercise and noise, and those that are actually physically active compared to age-matched controls have higher entropy measures, all things being considered. So there's something to it, which is the details just to be worked out. I'm quite optimistic this could be quite a useful measure. It may not be the answer, but it would be a way of actually being able to assess efficacy of different interventions.
And like with young people, what about ADHD in terms of noise?
Yeah, there was a group in Boston Children's Hospital actually applied that to a cohort of ADHD kids. And what they found was that the scale dependency I showed in the older adults, you see that in ADHD kids, but it's the other way around.
Yeah, that's right.
Did tone indicate
Did
tone of the noise? Because you did introduce some external noise with this, didn't you? In your
here, no, this is just measured in the
Oh,
brain.
all right then, okay. But I often wonder about tone with stimuli, because at a certain point, it just becomes too much, right?
Yeah, yeah. And that's why you have this nonlinear function. If there's
Yeah.
not
Yeah,
the kind of noise is quite important.
yeah, exactly, yeah.
And when I talked about the models, for instance, the noise that was introduced there was just this sort of broadband white noise. But in actual fact, the noise that's in the brain that's best is this, what's called 1 over F noise, which if you plot it has this high peak at low frequencies and then drops off gradually. Whereas white noise is very broad spectrum. Across. That 1 over F noise is an index of healthy biological tissue.
One of our viewers has a question. She's curious to know if the structural functional variability of the brain is or can be influenced by variability dimensions of culture, such as lifestyle issues of diet, exercise, family interactions, and general quality of life.
Sure. I just mentioned, for instance, that exercise will influence these variability measures. It's perhaps not that surprising if you consider that cardiovascular health is going to influence brain health as well. So definitely anything that's going to impact brain health is going to impact the variability measures. One nice thing about this metric, and this is kind of tooting my own horn, is that it can be measured very quickly, very easily, and it's much more sensitive than any other measure that you use in the brain. So typically what people will do with brain imaging data is look at average activity. You first of all need a lot of data to get that measure, and second of all, it's not very sensitive. Whereas this variability measure probably really embodies what the brain is actually doing. Because we're typically moving across time, right? Behavior goes forward in time. There's variability in what we're doing. We don't average our behaviors. We're doing things differently at the time. And that variation is quite important to capturing brain dynamics, and we can measure it with these tools. So yes, I think these other dimensions of culture, nutrition, and exercise will definitely impact these dimensions. Good. Thanks, everybody. Enjoy the rest of the day.
Randy, you're really in the sweet spot. And we really appreciate it, so we're giving you our little mug. Because we're really broke, and we hope you
That's okay. Thank you.
appreciate
We
it.
can always use that.